Dear students, in today's lecture, we'll discuss about the bacterial growth requirements. Now, before understanding the different requirements of bacterial growth, first we'll understand about what do you understand by the bacterial growth. Now, if we talk about the growth of a bacteria, here basically we are talking about increase in the number of bacteria. It is not related with increase in the size of the bacteria. As we have discussed in the earlier class also, the size of the bacteria ranges from 0.75 to 4 micrometers in diameter, but still if we are talking about the growth of the bacteria, means it does not mean that we are talking about increase in the size of the bacteria, but in growth here actually means increase in the number of bacteria. Okay, so the next point is why the growth requirements are studied. This is again a very important thing that we need to discuss before understanding the different growth requirements. So this can be understood in such a way, like for example, if you are performing some laboratory experiment, microbiology experiment, in which you are studying the identification or isolation or characterization of particular category of a bacteria. So to perform those experiment, actually we need some amount of microorganism and say for example bacteria. Okay, so if the number of bacteria at the initial culture is very less, then definitely we have to go for increase in the number, so that at least we have such amount of some amount of bacteria that can be utilized for performing that particular experiment. So initially, for example, if we are having a very less number of bacteria, then we need to study the different growth requirements that can favors the growth of the bacteria, so that at least you can have the amount which is actually required for performing various microbiological experiments in the laboratory. Similarly, if you are talking about certain pharmaceutical industry, say for example, if you are producing the product like antibiotics, vaccines, monoclonal antibodies or recombinant proteins, so definitely in those industries, the number of bacteria plays a very important role, because the industry think that if we can increase the number of bacteria, then definitely more amount of product will be generated. So in that case, again we have to go for the optimization of various growth requirements, so that we can produce the maximum number of bacteria. So again, coming to the thing, like here two things are very clear. First, if we are talking about the growth, then definitely here we are concerned about increase in the number of bacteria. And secondly, the study of growth requirement is very important, because there are certain experiments, there are certain production houses, they actually depend upon the growth or increase in the number of the bacteria. And initially, once we isolate a particular strain of a bacteria, then we have to go for its isolation, characterization type of studies. Then in that case we need certain amount of bacteria. So that particular microorganism or bacteria has to be cultivated in the laboratory. Now, coming to the next point. The bacterial growth requirements are actually divided into two categories. Number one is a nutritional requirement and number two is a physical growth requirement. So in this class, we'll study both the categories of growth requirement. First, we'll deal with the nutritional growth requirement and secondly, we'll deal with the physical growth requirements of the bacteria. Okay. So coming to the first point, the nutritional requirement of a bacteria. So in first we have to understand nutrition means what. So in the later class we'll also discuss like for the cultivation, for the growth of a bacteria, different categories of nutritional medias are available. Okay, so these nutritional medias provides different categories of the nutrients, including carbohydrate, proteins, lipids, and all category of the nutrients which are actually required for the growth of the bacteria. So again, if we have to cultivate a particular bacteria in the laboratory, first we have to understand, so for the growth of that particular bacteria, some minimum nutritional requirement are necessary. And if we can provide those things in the culture media or in the growth condition, then definitely the number of increase in the number of the bacteria will be very high. Okay, so here first we'll discuss about various nutritional requirement on which the bacterial growth is dependent. So the first nutritional requirement is water. Definitely, water is very essential for the sustenance of life, and life is not possible without the bacteria. And it is true for all the categories of the living organism, and same it is true for the bacteria also. In the last class, I have discussed if we talk about the cytoplasm, the 89% of the content of the cytoplasm is actually water. So definitely, if you are planning to cultivate a particular category of a bacteria, then first of all, the first thing that we have with us is water. Okay, so the first growth requirement is water. Now the next growth requirement is oxygen. Okay, because oxygen is again a very important thing on which the bacterial growth is dependent. So broadly, the bacteria can be classified on the basis of the requirement of oxygen. These bacteria are classified into two categories. One, the bacteria can be aerobic in nature, and number two, it can be anaerobic in nature. Okay, but again, if we go into the details, then we'll find out that these oxygen requirements are further divided into different categories. Okay, so one by one, we'll discuss all the different categories depending upon the oxygen requirement. The first category is obligate aerobes. Obligate aerobes are those categories of the bacteria which necessarily require the presence of oxygen. So they are entirely, the growth is entirely dependent upon the concentration of the oxygen. If the concentration of oxygen is less, then definitely growth of the microorganism or the bacteria will also be very less. Okay. So the next category is obligate anaerobe. Obligate anaerobes are those categories of the bacteria which do not require the presence of oxygen, and it has been observed that if the oxygen is available, then the growth of those particular bacteria reduces. Now, the third category of the bacteria depending upon the oxygen requirement is facultative anaerobes. Facultative anaerobes are those categories of the bacteria that can survive in the aerobic condition also, and that can survive in the anaerobic condition also. But more growth is observed under aerobic condition. So there are certain categories of the bacteria that can be cultivated in the aerobic condition also and under anaerobic conditions also. So those categories of the bacteria come under the category called as facultative anaerobes. The next category of the bacteria depending upon the oxygen requirement is aerotolerant anaerobes. Aerotolerant anaerobes are those categories of the bacteria which are actually anaerobic in nature, but they can tolerate some amount of oxygen. Like if we talk about the obligate anaerobes are those bacteria in which the growth occurs in the absence of oxygen, and if the oxygen is available, the growth of the bacteria reduces. But if we talk about the aerotolerant anaerobes, these are the categories of the bacteria which are basically anaerobic in nature, but they can tolerate some amount of oxygen. The next category of the bacteria depending upon the oxygen requirement is microaerophiles. Microaerophiles are those bacteria which are actually aerobic in nature, but they require a very small amount of oxygen. If the small amount of oxygen is also available, they can survive. And again, if we compare it with the obligate aerobe, then as I have told, the growth of the obligate aerobes depend upon the concentration of the oxygen. More will be the concentration of the oxygen, more aerobic growth will be observed. But if we compare it with the microaerophiles, these microaerophiles are the bacteria which are actually aerobic in nature, but their growth is not depending upon the concentration, and if the small amount of oxygen is also available, then these bacteria can easily survive. Now coming to the next nutritional requirement, that is a carbon. Now carbon is again a very essential nutritional element, and we can see like major of the functional components of a bacteria, they are actually made up of carbon. So bacteria can utilize the carbon in two forms. Number one, the bacteria which are chemoheterotrophs in nature. These are those bacteria which can utilize the carbon from the organic sources. Like they can utilize the carbon from the lipids, they can utilize the carbon from carbohydrates, from the proteins, etc. So whatever the organic matter is available, these bacteria, these are the categories of the bacteria that can actually consume that carbon from the organic sources. So these categories of bacteria are called as chemoheterotrophs. The next category of the bacteria depending upon the carbon requirement is chemoautotrophs. Chemoautotrophs are the categories of the bacteria which actually is not depending upon the organic sources, but rather they can utilize the carbon directly from carbon dioxide. Now, coming to the next nutritional requirement, that is a nitrogen. Nitrogen is again a very important nutritional requirement, and around 12 to 15% of the dry weight of bacteria is actually made up of nitrogen. So nitrogen also plays a very important role in the body of a bacteria. If we look at the structure of the DNA, if we look at the structure of RNA or a protein, then we'll find that these actually structural elements contain nitrogen. So for the survival of a bacteria, apart from oxygen, apart from carbon, nitrogen is again a very essential nutritional element. Now, nitrogen can be utilized by the bacteria again directly from the organic sources like protein, but there are certain categories of the bacteria which actually fixes the nitrogen from the atmosphere. Like their bacteria like rhizobium, bradyrhizobium, they have the tendency that they can directly fix the nitrogen from the atmosphere. Now coming to the next nutritional requirement, that is the tracer elements like iron, copper, zinc, molybdenum. These kind of the tracer elements are also required. They actually act as a cofactor for catalyzing various enzymatic reaction inside the body of a bacterium. So in a very small amount, these type of tracer elements are required. The next nutritional element is organic growth factor. Definitely vitamins, like as in case of human, we also require a little bit concentration of a vitamin. So is the case with the bacteria. Bacteria also require a small amount of vitamin. Let us discuss the physical growth requirement of a bacteria. The physical growth requirement of bacteria are basically divided into three categories. One is the requirement of a temperature, second is the requirement of a pH, and third is the requirement of the osmotic pressure. So one by one, we'll discuss all the three physical requirements which are necessary for the growth of the bacteria. Now coming to the first requirement, that is the temperature requirement. Now before understanding about the temperature requirement of a bacteria, first let us discuss three terminologies which are related with the temperature requirement. Number one is the minimum required temperature, second one is the maximum required temperature, and the third one is the optimum required temperature. Now if we talk about the minimum required temperature, it means at least this much amount of temperature is required for the growth of the particular category of a bacteria. And if you talk about the maximum growth temperature requirement, so it is exactly the amount of maximum temperature that can be tolerated by the bacteria. But if you talk about the optimum temperature requirement, it is actually the range of the temperature in which the growth of the bacteria can take place at the maximum numbers. So again, on the basis of the temperature, the bacteria can be classified into three categories. Number one are the cryophiles, number two are the mesophils, and the number three is the thermophils. If we talk about the cryophiles, the minimum temperature requirement is maybe it is less than 5 degrees centigrade, and the maximum temperature requirement is between 19 to 22 degrees centigrade. But again, the optimum temperature requirement for the cryophiles is between 15 to 18 degrees centigrade. The next category of the bacteria, depending upon the temperature requirement, are the mesophils. The minimum temperature requirement for the mesophils is 10 to 15 degrees centigrade. The maximum temperature requirement is 35 to 47 degrees centigrade, and the optimum growth temperature is between 30 to 45 degrees centigrade. And it has been observed, like if you talk about the industrial microorganism or the maximum number of microorganism, so maximum number of microorganism come under the category of the mesophils. Now, the third category of the bacteria, depending upon the temperature requirement, are the thermophils. The minimum temperature requirement of the thermophils are 40 to 45 degrees centigrade, and the maximum temperature requirement that can be tolerated by this category of the bacteria is between 60 to 85 degrees centigrade. But again, the optimum growth temperature required by the thermophils is between 55 to 75 degrees centigrade. So if we are talking about the growth, if we have to cultivate a particular microorganism or a bacteria, then we must focus on the optimum requirements. Actually, for a particular bacteria, there is already a set minimum temperature value and the maximum temperature value. But for the optimization study, the most required temperature, most studied temperature is actually the optimum temperature. Now, coming to the next physical requirement, that is of the pH. So it has been again observed, like most of the organism, they survive, they grow maximally at the pH between 6.5 to 7.5. But again, some exceptions are definitely there. Some bacteria can tolerate the acidic environment also, and some bacteria can tolerate the basic environment also. But maximally, the majority industrial microorganism, they are having the pH range between 6.5 to 7.5. Now, the third physical requirement is the osmotic pressure, that is the concentration of the solute. It is again a very essential criteria when we are optimizing a particular culture medium of a bacteria. So it has been observed in these studies, like if we keep the isotonicity in the solution or in the culture media which is required for the growth, which will be used for the growth of the bacteria, then definitely the maximum growth is observed. Again, if the solution is hypertonic in nature or hypotonic in nature, then definitely in both the conditions, if it is hypertonic in solution, then what will happen? The water will migrate from the region of the lower concentration of solute to the higher concentration of the solute, and definitely the cell will shrink. Similarly, if you talk about the hypotonic solution, the water will migrate inside the cell, and the bacterial cell can burst. So, the best suitable tonicity, if we talk about the growth of the bacteria, is actually the isotonic solutions. Thank you.